All right, we'll go ahead and get started. I'm sure some more people are going to trickle in here once we finish up this break. But really excited to have you all here. This is a new track this year. We realized that we kind of been ignoring ecology and wildlife for the last few years of the Iowa Water Conference, and it doesn't make any sense for us to do that, since obviously it's such an important part of water management in Iowa. So we're going to start, and then my computer with the bios, here we go, just give it a second. We have Jackie Gautsch and Brandon Harland from the Iowa DNR. We have Riverine Wetland Monitoring. And then now Jackie graduated from the University of Wisconsin. I'm doing this from memory because my computer is still in a second. I'll back up. Brandon graduated from Iowa State and also South Dakota State University. Jackie's been with the DNR since 2000. Brandon's been with the DNR since 2004. And they both work with the Shallow Lakes and Wetland Monitoring Program.
Good memory.
Thanks.
All right, thanks. And glad to see you guys here today. And I'm going to do most of the talking, and Brandon's going to answer all your questions at the end, okay? That's what we agreed to, right? How do I make it from there, folks? Okay, so just a little bit of background. You've probably heard this figure before, but over 90% of the wetlands in Iowa have been lost. The wetlands that we do have been dramatically altered. They don't function as they historically would have functioned in a natural system. Their hydrology has been altered. They have been disconnected from their floodplains, in particular with the riverine wetlands. They have berms. They have water control structures. They've been dredged into ponds. They have had fill put in them for when we do stream improvement projects. We, not as in me, but we as in Iowans. They have altered nutrient cycling, and there's lots of nutrients that are introduced, and so that increases the succession rate from the riverine wetland to the wet meadow to the forested floodplain succession. Okay, so this project that I'm going to talk about today is from 2012 to 2014. We have been doing a riverine wetland monitoring program in Iowa. So this is a statewide program, randomly selected wetlands throughout Iowa to look at the physical conditions, the water quality, vegetation communities, and the wildlife use. Historic information is basically non-existent for this class of wetlands. And we just wanted to improve the quality and the quantity of data available for these resource managers that either own these properties or manage these properties. And this project was funded by a grant through the EPA. Okay, site locations. As I said, the sites were randomly selected throughout Iowa using the WMI wetland mapping inventory layer. And the sites were selected so that we would have semi-permanent and permanent sites. This kind of varied. Most of ours are probably more towards the permanent sites just because a lot of the semi-permanent riverine wetlands have either been disconnected from their floodplains or drained. And they have to be located within 2 miles of a stream. That makes them riverine. And we targeted 20 to 30 wetlands each year. And then we targeted the first year to pick 25% of those to be sampled all 3 years. And so then we started getting permission and checking out sites. So here were the sampling locations that we ended up with, and you can see we have pretty well statewide distribution. We have more common wetlands in some of the areas, so the areas like the Wapsie or the Cedar or the Iowa River have more wetlands than, say, the Nishnabotna or some of our southwestern rivers, so you can see we have more concentrations over there. We do have a couple sites on the Missouri River floodplain. All right, so our monitoring consists of basically 3 different components. Chemical monitoring, so the water quality monitoring sampling, that happened 1 time each summer in May or June. Biological sampling, and so that was invertebrate sampling that happened 1 time during June or July. Fish surveys that happened 1 time, usually during July, and veg surveys that happened in July and August. Okay, the water quality for the chemical and physical monitoring, we collected water using a hand dipper to try to not stir up the bottom. You can see see a lot of these wetlands have very mucky bottoms. It's very unconsolidated material. You would walk in there and there would be a plume. So we try to use a dipstick so that we're not impacting our sample. We also use a turbidity meter to measure turbidity, and we used a YSI meter to collect dissolved oxygen, pH, and water temperature data. Then our water samples were sent to the State Hygienic Laboratory for analysis, and we tested for total phosphate, orthophosphate, forms of nitrogen, chloride, chlorophyll A, suspended materials, dissolved materials, and then we did some acid and standard herbicide pesticide sampling. Okay, invertebrate sampling. So we collected invertebrates once a year at each of these wetlands if they had water at the time. And we used 10 1-meter-long D-net sweeps. 1 year we collected samples both in the spring and the summer to see if there was a difference, and then subsequent years we only did one sampling event. We combined all 10 1-meter sweeps into one composite sample, and then they were identified, counted, and some indexes of biotic integrity were calculated. All right, fish sampling. Typically we use 2 different modified fipe nets with different mesh sizes to collect different size species. We also use some minnow traps. We allowed these to soak in the wetland for 24 hours overnight. And we sampled wetlands that were over 1 meter deep on average for fish. Okay, vegetation surveys. We conducted vegetation surveys at each wetland that still held water at that time in 1 meter plots spaced every 10 meters apart. So basically we would walk transects, and every 10 meters we would do a 1 meter plot, identify the plant species there, and rank the canopy cover for each species. Okay, so I'm going to talk about some of the data. We are still analyzing a lot of this information. So this is kind of just a short, brief overlook at some of the data that we collected. So turbidity, most often turbidity was pretty low throughout our wetlands. There were some wetland sites in 2012, and that's the blue sites, that had pretty high turbidity. 2012, those sites had very low water, had animal watering that was currently happening, and it was a very dry year that year, if you remember. And so the low water, along with animal use in those wetlands, typically ended up in higher turbidity. Okay, dissolved oxygen levels. There was one wetland in 2012 that actually suffered a fish kill in early August. And when we sampled the dissolved oxygen in that wetland, it was very low, 1.62 milligrams per liter. The water quality standard for warm water streams in Iowa is 5, and that's kind of a good benchmark to look at for whether or not fish and other invertebrates that use dissolved oxygen are going to be able to survive in that system. We also had a low dissolved oxygen level in 2013 that wetland was completely covered in lesser duckweed. There was no submerged vegetation in that wetland, so there wasn't a lot of oxygen influx happening there. Dissolved oxygen gets into water by mixing with the air, so turbulence can enter dissolved oxygen into the water. You don't have a lot of that in wetlands, so we mostly rely on the aquatic plants to produce oxygen for those wetlands species. In 2004, we had readings all over 7.1. So 2012 was very dry. 2013, we had a very wet spring. In 2014, we had a wet spring / early summer. All right, so nitrite conditions. 2012, like I said, was the 19th driest and the 3rd warmest Iowa on record. And so high drought and high evaporation rates led to very low water conditions. And so we found lots more nitrates in our systems. 2013 was one of the wettest springs on record. We had widespread flooding throughout our sites. And in 2014, it was the wettest June, or the 3rd wettest June on record. And again, we had widespread flooding, so that probably greatly affected our nutrient levels. Okay, phosphorus levels. Most of our wetlands are hypereutrophic systems, kind of to be expected. They're highly productive. There's lots of nutrient flux there. Usually there's lots of biological activity that would be happening in our wetlands to use up a lot of that dissolved phosphorus in our systems. Wetland 5A in particular, you'll see has a 2013 high record, and you'll remember that wetland because there's a couple other parameters that we found high levels of. Okay, so pH was pretty consistent in Iowa. We have a nice buffered soil in Iowa, so we don't see a lot of flux there. However, chloride, wetland 5A, again, you'll see a high chloride level. Chloride is kind of a conservative tracer. Once it shows up in the natural system, not a lot of things use it or break it down, and so it will hang out for a long time. All animals, including us, consume salt. Road salt is another contributor. Also, soil can contribute chloride in the environment. Okay, pesticide and herbicide detections. We found 14 different pesticides in our sampling. You'll see that 2013, 2014, we had more detects, and of course that was the wetter spring times. The most commonly detected pesticide is going to be atrazine. So atrazine in 2012 showed up in 12 of our 21 wetland sites. In 2013, it showed up in 32 of 34, and in 2014, 26 of 28. However, we had actually pretty low median concentrations of these pesticides. And again, we really think this is rainfall driven. Drought season in 2012, wet times in 2013-14. Okay, invertebrates. So the aquatic organisms that live in the wetland, these are very important for fish and waterfowl that we're utilizing these wetlands. And you can see that taxa richness in 2013 was higher. 2014 shortly followed that. And the same thing with abundance. And I'll say these are only the sites that we monitored year after year. A lot of this data we haven't totally analyzed yet, so we have a lot more data to look at. So we might have more interesting things to find. Okay, we calculated a couple invertebrate IBIs, an index of biotic integrity. So an index of biotic integrity is looking at a bunch of different measurements within that biological data. So you can look at habitat use, or you can look at pollution tolerance levels, or you can look at predator-prey or food feeding guilds, that sort of thing to calculate an IBI. This particular IBI was developed in Minnesota, and it looks at specific wetland species, things that you would see in a productive, healthy wetland. So things like dragonflies and damselflies, leeches. It looks at how many beetles compared to how many true bugs or water boatmen, back swimmers that you find. And so you can see that the line that is orange, anything below that is going to be a poor rating for this IBI. Anything between the orange line and the purple line is going to be a median value. It's going to be a moderate. And then anything above that purple line, which we don't happen to have any, are going to be excellent populations. So you can see that Iowa, when we're looking at this IBI, isn't rating so great for these riverine wetlands. Okay, so individually looking at 2012 for that Minnesota IBI, you can see all of our levels, all of our species were in the low range, or our index was low. And again, talking about the rainfall, we think that probably influenced. Gets a little better. For 2013, there was one site that was in the excellent category, quite a few that were in the median, and then again, we had quite a bit that were in the poor range. 2014, again, we didn't do so hot in the Minnesota IBI. The thing about riverine wetlands and their altered hydrology is that a lot of the species don't necessarily have a terrestrial form, and so when they're coming into these wetland systems, they are coming in through that connection to the river. So the floodplain and the river connection is very important for repopulating these species. So things like water boatmen and scuds and sow bugs, they don't have an adult terrestrial form. They spend their entire life in the water. Whereas things like midges and mayflies and dragonflies, they have a terrestrial adult form, so they can travel between wetland sites and repopulate areas. Okay, a little bit about the fish sampling. In 2012, By the time we got to our fish sampling, we didn't have a lot of water, so there weren't as many sites that we were able to fish net. There were 2 wetlands that did not have fish that we sampled, and there were 4 that we did sample that had fish. We only found 5 species that year. 2013, 1 wetland that we sampled did not have fish, 9 of them did, and we found 22 species. Okay, wetter year, more connection to the floodplain, more connectedness, more influx of species. 2014, all 20 wetlands that we sampled, we found fish in, and we found 35 different species. Some of the common fish that we captured are bullheads, minnows, carp, bluegill, green sunfish. These aren't always necessarily highly desired fish. A lot of them are fish that might stir up the bottom, create turbidity, reduce the amount of aquatic vegetation that can grow there, and therefore that's going to impact the vertebrate population as well. Okay, a little bit about the herps, the amphibians, and reptiles that we found. Lots of turtles were found in Iowa wetlands, if you can believe it or not. A couple of significant, we found a spiny soft-shelled turtle in one of our wetlands, and we found a couple of wetlands that had gray-winged turtles, which is an endangered species in Iowa. Okay, vegetation survey. So the number of veg species that we found in Iowa did vary a lot per year. There were some wetlands that had more fish populations, had more turbid water, had less vegetation, and then there were the wetlands that were either didn't have those fish that stir up the bottom, that don't disrupt the plant communities, and had open canopy covers. Sometimes our forested wetlands, it's harder for that submerged aquatic veg to grow. We didn't find as many species. We did, however, find invasive species. We found, on average, about 2 invasive species in those wetlands that had invasive species. Commonly, reed canary grass, creeping charlie, curly pondweed, and honeysuckle were those invasives. Okay, a couple noteworthy collections and observations that we made. We did find a state endangered species in a wetland in Alameda County, swamp loosestrife. Has pretty flowers. Is related to the invasive loosestrife species, but this one is native. Water fern was found in a wetland in Wisa County. We also found them in 2014, I haven't gone through all that data yet, in a wetland in Jackson County and one in Clinton County. Blanding's turtles, as I talked about, were found in 2 wetlands, one in Mitchell County and one in Lynn County. And then also one wetland in Wisa County had a very dense population of rhizoans, freshwater rhizoans. This is a colony-forming organism, and they're indicative of clean water. And we also did in 2014, this was in 2013, 2014, the wetland in Jackson County also had some of these species. So, they're kind of cool. Okay. Does anybody have any questions for Brandon?
You had wetland, it was like 24, that was an excellent diversity for vertebrates. Do you remember which one that was?
Wetland 24.
Pretty sure it was 24. Yeah.
In what year?
2013.
It is going to be in southeast Iowa. I don't tell you that much. We're currently putting together a report with all this information, and it's going to be on our website. So hopefully we'll be able to get more precise information out there to you. But if you email me, I can tell you where it is. Yeah.
Regarding the pesticide levels, did you have any kind of benchmark that you were comparing those numbers to, or just to kind of give us a context of, are these levels concerning? Are they at levels harmful to organisms, or?
They, so far from the data that I have looked at, and that's just the revisit sites, they are found in pretty low concentrations. However, I have not looked at whether certain degradates or certain pesticides, whether the acute or chronic toxicity levels have been reached yet.
Okay.
So I haven't gotten that far.
Okay.
Were the Blanding's turtles populations disjunct from known populations, or were they close to known populations of Blanding?
Both were found along the Wapsie River watershed. So that's a pretty known population one, the location was already a confirmed sighting, so.
You said that the species of rhizome
Mm-hmm.
was an indicator of good water quality?
Yeah.
Are there others like that you were able to, I mean, was there anything else besides the rhizome that could also indicate water quality?
So for that wetland is kind of a highly altered hydrologic wetland, the one in Louisa County. It has a large berm around it, so it isn't necessarily very connected to its floodplain. So the invertebrate population that were found there weren't as high. So I was kind of surprised to see the rhizome in there. Not in that particular wetland. I can't find any others. So maybe.
I was curious if you could share some of your thoughts about similarities and differences between the riverine wetlands you looked at and then some of the other constructed wetlands that we're seeing a lot of around the state. I mean, probably differences in fish for sure. What other similarities or differences would you expect in terms of the biotic integrity?
It's hard for me to say for the constructed wetlands. We haven't really done a lot of monitoring, looking at the biological communities there, the plant communities. When restoring some of these wetlands, like the prairie pothole wetlands, if you have the natural system there and fish have been introduced, you can remove those fish and draw down the water and plants will grow back. The wetland plants will. A lot of the constructed wetlands, they have to plant actual herbaceous live adult plants or seed it. And so I think there is some flux there on how quickly those populations rebound and the biology reflux from that. We haven't done any direct comparisons. Riverine wetlands are not necessarily a very common restored wetland either. Prairie potholes are going to be more common.
What's the plan, EP's plan for the data? What are they?
So this data was kind of collected for baseline information to find out what is the quality of our riverine wetlands. We have done projects on prairie pothole wetlands. So this next summer, in the next couple summers, we're going to select sites from this subset to be reference sites. And so we're going to look at prairie pothole reference sites, fen reference sites, and riverine wetland reference sites and collect data on that.
Have you found stability in the biological communities and the difference of the sizes of the wetlands from the riparian?
That's a good question. I think I found more connections between floodplain connectivity. So if the wetland hasn't been highly altered, it doesn't have a berm or a levee between it and its river, we've seen higher, at least I have, invertebrate populations. However, then you also get increased fish. And some of our rivers have depressed fish populations, so you're finding things like carp that are more common and bullheads and things like that get into the wetland then, and then that can be a negative impact. So there's positives and negatives to being connected to your river system still. But wetland size I haven't really seen. I haven't looked a lot at that either, so that might be something interesting to investigate. We have. In fact, years ago, when I was more connected to the iWater program, we did develop a protocol for wetland monitoring. And so I'm hoping that that would be something that we could do, incorporate more volunteers with doing wetland monitoring, even if it's just looking at some of the biota or the plants, plant systems, as opposed to taking lab samples. I think it would be fun. If anybody wants to come out and monitor with us this summer, I would love to have a volunteer tag along with me, so that would be cool too. Anybody else?
I've got somebody for you. So.
Okay, perfect.
They're not here.
You're gonna volunteer them. You're gonna.